Now, what is a capsid?
The capsid is the
Capsid is a protein shell. What are the three different shapes a virus may have?
lot.
Mention them.
Mesogenic?
Helical.
I don't know.
He mentioned it.
Really?
Yes. It's helical,
Many-sided?
many-sided, or envelope. Envelope. Right, insoluble. Is a bacteriophage a virus or a bacteria? Explain.
It's a virus.
Why do you think so?
Because it invents bacteria.
It infects the what? Infects bacteria. Is that the reason why it's a virus?
Yes.
Another reason? So virus that infects bacteria. What type of infectious pathway is associated with the prophage or provirus? Prophage.
Associated with the prophage.
Yeah, I'm asking you.
Yeah, I said the answer is prophage.
So what type of infectious pathway is associated with the prophage or provirus?
Prophage.
What do you mean prophage? Prophage is the name of a phage, a bacteriophage.
Even the virus? Provirus?
What do they mean by provirus? Before the virus become formed. So here they're going to form a what? Lysogenic, because it's the time before the viral replication. Well, here they form what? A lytic phase, because after the replication, they need to find a way to come out of the cell. At that point in time, we say they have a lytic cycle. Some viral diseases can be prevented with vaccines. Okay, let's look at them and talk about them a little bit. Viruses cause many infectious diseases. They're very deadly disease. There are many examples of viral infections. Common cold is an example of viral infection. This is the way it forms around the nasal region. Influenza is also a good example of viral infection. And SARS is also an example of it.
I have a question in the book.
Okay. HIV, the full meaning of HIV is human immunovirus. So this is the way it looks. HIV infected white blood cell. So the white blood cell is already infected with the HIV virus. So it destroys the white blood cell and make the immune system very weak. So that's why I told people that when people talk about HIV/AIDS, what actually happens is the virus just infects the immune system and weakens the immune system. It's not supposed to be it's not supposed to be a big disease that you're saying is incurable or we can cure it. Because the immune system is weak, okay, just help the situation, fix the immune system. So HIV, the body has natural defenses against viruses. So vaccines are made from weakened pathogens. Let me explain this to you. How do we make vaccines? We make vaccines by attenuating, because something attenuation. Attenuation means we're weakening the pathogens, the causative organism. The causative organism of a disease, we will weaken it. So vaccines stimulate the body's own immune system. What do I mean? The meaning is the causative organism, what causes the diseases, the viruses, they would be killed a little bit. They will kill them a little bit, but called attenuation. So now when they weaken, then they will put them into the body of the host. So the body's immune system will be used to kill them because they're already weakened, right?
Yeah.
So there's something we call remembrance in immune system. the immune system has a capacity to remember what it killed before. So, and it's going to prepare more to kill it when it sees the thing again next time.
Really?
Yeah. So vaccines prepare the immune system for a future attack. So it's called remembrance. So these are examples of viral infection, which we have chickenpox. Among the symptoms of chickenpox is rash, itchy skin, fever, fatigue. How does it get to the place? Contact with rash, droplets. Let's say, for example, someone has got chickenpox next to you, and it comes to school, you use the body to touch his body, immediately you're going to have it. So it's actually used for children under between 12 to 18 months. Hepatitis A, you're going to have a yellow skin, fatigue, abdominal pain. So the way to get this is the contact with contaminated feces for people traveling to infected locations and protection during outbreaks. So in the US vaccine recommendation, they're going to give you these vaccines if you travel into places where there is a high spread of hepatitis A. MAPS is a painful swelling in the salivary gland or fever. The salivary gland will swell up, it's going to become so big. How can you get this infection spread? Droplet inhalation. If someone sneezes, that's why whenever you sneeze, try to cover up properly. If someone has got mumps and he sneezes openly, he can easily infect other people. So it's meant for children between 12 to 15 months and again 4 to 6 years. Rabies is an anxiety, paralysis, fear of water. That's what we call rabies. So the bite from an infected animal, if you have an infected animal which is infected with rabies, if you bite somebody, the person is gonna have rabies. Rabies is actually a disease which is transmitted by animals which actually have it. If, let's say, for example, monkeys, dogs, cats, they have rabies and they bite people. So you will notice that there will be paralysis, anxiety. There will be fear of water. So it won't want to, it won't want to pour water on the body. It's gonna run. Some kids are like that. They don't want to take shower. So, but sometimes these are the symptoms we see when we know someone has got rabies. West Nile disease is actually caused from what infected. When mosquito, which is already infected, when it bites somebody, then it's going to cause it. So it causes you fever, headache, and body ache. There's no vaccine for it too now.
What is body ache?
You're gonna feel pain on your body.
Where?
Body pain. Everywhere you feel pain in the joints, you pain in the legs, it's called body ache. Vaccines are the only way to control spread of viral disease. So this is all we have for this. Many infectious viruses exist. So the example of illness caused by viruses include the common cold. So we mentioned that before, right?
Yes.
So more than 200 viruses are known to cause a cold. More than 200. Can you imagine how much the number is? So now tell me, what kind of medicine does a pharmacist want? A pharmacist can work for 10 years before he's been able to get one medicine for a disease. He has to think. He has to do a lot of research, and
You don't need, sir. You can just give him the medicine by the doctor.
I mean, getting new medicine to form a new medicine.
New medicine.
You've been using the medicine, and the organism is getting resistant to it. So now look at it. Can you imagine? Now you've got 200 type of viruses. So how do you want to be able to know the one which is going to be effective on it? So this is why common cold, as simple as it is one of the deadliest diseases. It's not easy to treat. So with so many viruses, it is not easy to find a cure to it. In fact, cold viruses can mutate as a move from one person to another. If you have cold, you sneeze now, I get infected from you. It can change the shape and the size and everything before it gets to me.
That's smart.
Of course, they're smarter than you. So influenza is a flu, also spread quickly, can result in a frequent local epidemics. And it can spread within the community, everyone has it. What is an epidemic? An epidemic is a rapid outbreak of an infection that affects many people. Let's say, for example, someone has got a disease, has got influenza in school. In a class, if you have someone studying that has influenza in a class, everybody in that class will have it within 2 to 3 minutes. Oh, yeah, it spreads so fast.
That fast?
It spreads so fast, so the virus will spread. So this is the reason why if someone has got this viral infection, they say they quarantine the person. They tell them to stay away from people. So the rapid outbreak of infection that caused many people, the high mutation rate of flu viruses makes it necessary for a new influenza vaccine to be made every year. A vaccine, what's a vaccine? A vaccine is a substance that stimulates the body's own immune response against invasion by infectious agents.
Does stimulate mean?
It stimulates, it triggers, it makes your own immune system to affect the world, to kill the
does
world.
it work harder, yani?
You know, if yani, to stimulate means it makes your immune system know that it has to function.
Ah, okay.
Is that understood? I mentioned it to you. If a vaccine is used for you, the vaccine is actually used for you to make your body remember the same disease where they infect you. People with polio vaccine. So when someone has polio, it can go there. It's not going to be affected because the body system is already aware that there's a disease called polio and it already killed it before. So it's got the remembrance of how to deal with it. So once a person has already got a vaccine for a disease, he cannot have the disease again. So SARS is another one. It's called severe acute respiratory syndrome. It has symptoms similar to influenza, such as fever and coughing or difficulty in breathing. SARS is relatively recent concern. The spread was in 2002, I think, in China. It spread so fast, so that people were actually meant to cover their nose and stay back home because of SARS. So HIV, human immunovirus deficiency virus, or HIV, is a retrovirus. What does retro mean? Retro means backwards, which describes how retrovirus works. The retrovirus contains RNA and uses an enzyme called the reverse transcriptase to make a DNA copy. So there is a method we're going to take in grade 11. It's called reverse transcriptase. This is the way in which RNA forms a DNA. Is that understood? So in comparison, most viruses use DNA to make an RNA copy in the cell. So the meaning is most viruses, they actually use DNA to form an RNA copy. But an active HIV infection destroys the white blood cell and make the immune system very weak. So that's why it's being called a reverse transcriptase. It's going to produce what? It uses an enzyme called reverse transcriptase to make what? DNA. Is understood.
Yes.
It reverses the reaction to make DNA, whereas organisms are meant to split DNA to two to form RNA. Yeah. Organisms usually, all the viruses which are known, they split DNA to two to form RNA. But this is going to form a DNA from RNA. That's why they call it a retrovirus. Retro means it reverses the process. It reverses the process against what it used to be before.
Yeah.
What it used to be is that you're going to see what DNA will split to form RNA molecules, but this is going to form a DNA from an RNA. So, and when you've got a loss of white blood cells, ultimately cause what we call AIDS. So AIDS is actually caused because the white blood cells quantity or quality is very low. So the person is prone or exposed to a lot of diseases or infection. Is that understood?
Yes.
So once a person's immune system is affected, he or she may be unable to fight off even the common microorganism that humans encounter every day. Vaccines can prevent some, but not all viral diseases. A vaccine is made from the same pathogen because, pathogen disease-causing agent that is supposed to protect against. Is that understood?
Yes.
So we form a vaccine from the same pathogen. How do we form this? We're going to kill it. We're going to weaken it. We call it attenuation. We attenuate it. So when it becomes weak, then we inject it into the person's body. So when it gets to the body of the person, then it makes the immune system to fight it and kill it very easily. So the immune system would remember that same disease, the same pathogen, whenever they enter the body again. So that's why the person will not be affected by the disease in the second time.
So if we had influenza, your sore throat,
Yeah, cough,
cough, yeah,
inability to breathe.
always comes.
Yeah.
How does the immune system does not remember أصلا?
Oh, because they don't have a vaccine for it. You've not been given a vaccine for influenza. Is understood. So the vaccines consist of weakened versions of viruses or part of the virus that will cause the body to produce a response to it. In the host body, the vaccine works by preparing the host immune system for future attack. Why is it hard to find a cure to common cold?
Because it's just small. There's no what's it called?
The shape.
The shape changes.
The shape changes frequently. How does receiving the vaccine affect the person's immune system?
How did it?
How does receiving the vaccine affect the person's immune system?
It makes the immune system.
It triggers the what? It triggers the immune response. It makes the immune system fight the what? Fight the disease. Why is HIV called retrovirus?
Because it just copies the DNA. Does not copy. I mean, does not split into
It
two.
uses reverse transcriptase.
Ew.
That's why I said transcriptase. The meaning is it forms a what? It forms a what? A DNA from a what? From a RNA. Whereas it's supposed to form what? RNA from DNA. Why might a mild infection act similarly to vaccine? How might a mild infection act similarly to vaccine?
Mild means small.
Small, little, not too much.
Because the pathogen is weakened. Because the pathogen is weakened?
No, because the number of the pathogen in the mild infection is not much. So the immune system will be able to come back and kill the pathogens. And the immune system would know that pathogen is already been there before. It's going to remember it. When it comes again, like a very full infection, it's not going to be able to infect you again. So when you have a mild infection, the mild infection can actually function as a vaccine because it's going to make your immune system remember the disease when it comes back to you again. Now, bacteria and archaea. Bacteria and archaea are both single-celled prokaryotes. They're both what? Single-cell prokaryotes. What do they mean? They have a single cell, then they don't have a what? No nucleus, right?
Yes.
And they have no membrane-bound organelles. No membrane-bound organelles. You know, the scientists, membrane-bound organelles. The scientists actually put the archaea and the bacteria together before, but later on they got to know that they're not the same. So prokaryotes are widespread on the earth. They're everywhere. Everywhere you go. Prokaryotes can be grouped by their need for oxygen. We can group them by their need for oxygen. Obligate anaerobes. Anaerobes are organisms that does not need oxygen. They're organisms that need no oxygen. That's the meaning of anaerobes. Oxygen. Obligate anaerobes are poisoned by oxygen. They don't need oxygen. That's why they're called anaerobes. Anaerobes means it doesn't need anaerobes. Doesn't need oxygen, no oxygen, right?
Yes.
So these are examples of the obligate anaerobes. We have obligate aerobes. They need oxygen. Aerobes means, is that not your question?
Yeah, that's the question.
So they need oxygen. They need O2 to live, to do anything they want to do.
But they can live without the oxygen.
They can actually live for a short time, depending, but the facultative aerobes can live with or without oxygen. Is that understood?
Yeah.
The facultative are the ones that can live with or without. They call them facultative aerobes. You know, aerobes, they need what? They need oxygen, right?
Yes.
But when they become facultative, they can live with or without. But obligate anaerobes, When they find oxygen, they get killed, they get poisoned. Obligate aerobes, they need oxygen. Without oxygen, they easily die. But the facultative aerobes, they can live in presence or absence of oxygen. Bacteria and archaea are structurally similar but have different molecular structures. Bacteria commonly come in three forms. We have a rod shape, which is called a bacilli. They look like this, like a rod. We call them bacilli or bacillus. So this is the way they look. Example of this is lactobacillus. Lactobacillus is the one that is being used to make your milk ferment to form yogurt.
Yogurt?
Yes, used to form yogurt. It's called lactobacillus. It's going to be put in the milk and it makes the milk to ferment. That's why the milk is sour when you drink it. So it's called lactobacillus. So it's one of the form in which the bacteria form. Another one, the spiral, which is called the spirochete or the spirula. They look like they look like this. You see them? They're connected like this, but the small, tiny, like ribbons. Because they call them spirals. Example is spirochete. Another one is spherical, which is called cocci. So the cocci looks like this. It looks like a sphere. Did you see that?
Yeah.
It looks like it's called spherical. Example, this is enterococci. So all of them cause disease. They cause diseases for people, and they're very deadly. Or archaea have many shapes too. Remember the shapes of bacteria? Can you tell me again? Bacillus.
Bacillus is a rod. The what's it called? The
Spirochete.
Spirochete spiral.
Spiral means small connected together, right?
Yeah.
What about the last one?
Enterococci is the spherical.
Spherical. They form like a sphere.
Yeah.
Bacteria and archaea have similar structures. They both have plasmids. They both have flagellum, and they have the pili.
What is the?
You're gonna know them. Now the flagellum is actually used for what? For locomotion. Locomotion means movement. Yeah, movement. The plasmid is what? It's just
plasma.
Like a genetic, not plasma. It's just like just like a genetic material which they have. It's called plasmid. So what about the pili? It's just like the hair-like structure they have on the surface called the pili. So the diagram shows the typical structure of a prokaryote. Archaea and bacteria look very similar, although they have important molecular differences. Now, bacteria and archaea have molecular differences. The amount of peptidoglycan within the cell wall between them is not the same. What do we mean by the peptidoglycan? We're talking about peptide and glycogen.
Yeah.
So peptide is got from where? And glycogen. Glycogen is what is sugar.
Glucose.
Yeah, sugar. Glucose is got from glycogen. What about peptide?
I don't know.
Glycogen. What is a peptide got from? Now the peptide from the glycogen or it forms the peptidoglycan. So the peptide is actually what? Proteins, or we say amino acid, and this is glucose. So these are the things, two materials that form the peptidoglycan, and they are the composition or the amount we have within the cell of a bacteria, and a archaea is not the same. Now, the very easy way we used to know bacteria when we were in the university is the gram-positive and gram-negative. When you say gram-positive, gram-negative, we're talking about the amount of peptidoglycan they have in the cell wall. This is the outer membrane of the gram-negative. This is the cell wall, and this is the cell wall of the gram-positive. Now, which of them has more peptidoglycan? The gram-positive, and this is a plasma membrane. So this is why sometimes when you stain them with safrani or methyl blue in the lab, you will see the gram-positive will have a darker color than the gram-negative because it's got more
more
peptidoglycan to be able to hold onto the stain. So archaea have different lipid entirely. The archaea have different lipid entirely. Now gram staining. Gram staining identifies bacteria. This is exactly what we use to identify bacteria. Stains polymer peptidoglycan. Gram positive stains purple. It has more peptidoglycan. So it's going to be darker in color because it takes more of the stain. So that's why it looks purple and it's darker when you stain it. But a gram negative, it looks pink because it's taking a little because it has less peptidoglycan. So whenever we stain bacteria in the lab, we use methyl blue and we use safranin. The safranin used to be the last stain, and that's what stays red or pink or purple. Yeah. We use the methyl blue to make it thicken to the slide. Then we're gonna wash it off. And we'll put the safranin. So the safranin has a reddish color. So it's gonna stick more to the gram positive and makes it become purple because it has more peptidoglycan than the gram negative. So now this is the way it looks, exactly the way it looks under the microscope when you stain them in the lab. Is that understood? Yeah, this is the way it looks. So you will stain it and you see it this way, you're gonna know it's gram positive or gram negative. So now bacteria have various strategies for survival. The prokaryotes exchange genes during conjugation. What do they mean? The bacteria can exchange gene with the archaea when they conjugate. Conjugation is a method of reproduction, which is an asexual reproduction method, which is being used by the bacteria and archaea to reproduce. They're going to have something to connect together, and they will transfer genetic material. And this is why many scientists say bacteria resistance to antibiotics is increasing every day. Because how would a bacteria exchange genetic material with an archaea? If it does it, then it's going to have a crazy gene and will not be able to be affected by antibacterial medicines again. So that's why the scientists are facing a big problem, a very big problem to find a solution to antibacterial resistance. Bacteria may survive by forming endospores too. Some bacteria form endospore. Endospore is just like a very hard shell, which the bacteria can hide inside. The bacteria will hide inside it, and the hard shell would not allow any medicine to have contact with the bacteria. So it's called endospore. So they can remain like that for years without breaking. So once they have access to a wet place, then the endospore will break open and they will start to affect or cause disease. They will also reproduce. So this is all we have for bacteria and archaea. So let's take a break. We'll come back later, inshallah.